Hello, this is my presentation on ACE2 transportation. The goal of this assignment is to learn about how angiotensin converting enzyme 2, also known as ACE2 protein, is transported to its final destination in lung cells. My name is Alexandra de la Cruz. So, specifically, what is ACE2? I want to be able to cover the techniques, the process of glycosylation, protein trafficking and transport, and of course, important details that I want to cover. So, some important techniques include mass spectrometry. This technique uses a combination of enzymatic proteases such as trypsin, glycopeptides, and other peptides as well. Another important technique would be called fluorescent dye staining. This is the use of fluorescent dye in order to identify a specific sequence and or protein. Now moving on to the process of glycosylation. What is this and what does it do? So, glycosylation is the addition of a carbohydrate side chains to a very specific amino acid residue of protein. This eventually will turn into a glycolipid, and this is known as the process of glycosylation. This process starts in the endoplasmic reticulum and ends in the Golgi apparatus. So, this is why the Golgi apparatus and the endoplasmic reticulum are highly important. There is two types of glycosylation that I will be discussing. They are N-linked and O-linked. Starting off with N-linked, this type involves adding a specific polysaccharide unit to the nitrogen atom. O-linked involves adding an oligosaccharide to the oxygen atom. So now moving on to protein trafficking and transport. When talking about protein trafficking, it's important to know that there's two important directions and components when it comes to protein trafficking. These include an anterograde and retrograde. Anterograde begins when the protein is synthesized in the ribosome of the rough ER. Following that, the protein will then enter the ER lumen where glycosylation is taking place. Moving on to retrograde, it includes proteins and other materials such as peptides and other material, of course. These materials are being moved and transported with the help of endocytosis. They're moving in and out of the cell with the help of endocytosis. Then they will form vesicles that mix and fuse with early stages of endosomes that contain important proteins that are vital. These will then mature and they will eventually turn into lysosomes. So some important details that I want to touch base on include the following. ACE2 is involved with N-linked glycosylation, and this takes place in the N-terminal extracellular domain. So now moving on to another bullet point that I wanted to touch base on is what is protein trafficking? So prior to this presentation, I did not know what it was. I myself had to educate myself, do some research, and educate myself as to what is it. So protein trafficking means that proteins will be sorted out to where they are destined to be. It's a mechanism that transport proteins to their acquired and appropriate destination. This could include both in and out of the cell. So I think of protein trafficking such as a traffic officer leading traffic to the right places, ensuring that there is no accidents being happened. So that's what I think of protein trafficking. It's just basically making sure and ensuring that proteins are where they are supposed to be, where they are destined to be, and if they are not, making sure to move them and make sure that they are in the right place. So now moving on to experimental designs with protein RSC. So a microscope-based experiment I would use is fluorescent dye. I would make sure to tag the protein and make sure that I can observe the fluorescent proteins in the microscope. So of course, with the use of the fluorescent dye, it is very bright. I would specifically see the sequence of the amino acid from the specific identified protein. Not only will it specify the proteins and the sequences, but you can see the immunoglobulins lacking, and you can see specific protein sequence. A control would not be needed because you have the entire help of the fluorescent dye helping you out. With the fluorescent dye, you can see everything. You can identify sequences, proteins, and more. So a non-microscope based technique that I would use, there's two. I would use centrifugation and biochemical testing. By using these techniques, you can easily identify and predict in detail the sites with the help of bioinformatics and statistical analysis as well. A control that you can use with this technique would be a unknown, undisturbed, and non-identified cell. So by using centrifugation, you can easily identify and predict in detail. And these are my citations for the picture. Hope you enjoyed.